This is the video on plasticity. Neuroplasticity, or brain plasticity, refers to our nervous system's ability to alter its responses to stimuli by reorganizing itself. There is short-term plasticity that relies on the modification of existing synaptic proteins, but it is in the long-term plasticity that we see new gene sequences give rise to new proteins that, in turn, make new synapses. Long-term potentiation, or LTP, is the type of plasticity that results in an increase of synaptic strength. Long-term depressions, or LDP, however, is used to describe decreased synaptic strength. Both are forms of synaptic plasticity. The hippocampus is the main focus for neuroplasticity in mammals. From neuroimaging, we know that the hippocampus is involved in many memory tasks. In the 1960s, researchers found that a high electrical stimulation of the hippocampus increased hippocampal synaptic transmission for hours. This is due to the efficient layout of the circuitry in the hippocampus. In just one slice, we can see all the cell bodies of the pyramidal neurons. The CA3 cells have axons called Schaefer collaterals, which play a large role in understanding LTP. Though a small stimulation of the Schaefer collaterals results in a proportional excitation of CA1 cells, above a certain threshold, the relationship results in LTP. When a high-frequency stimulus is sent to that region of the brain, the CA1 cells have considerably higher EPSP amplitudes for a prolonged period of time. This results in increased memory storage for that subject. The reason for this is outlined in this picture, and the mechanism is due to NMDA receptors. Magnesium blocks the NMDA receptors on the postsynaptic membranes at resting potential. However, when a depolarization of a high enough threshold occurs, magnesium is expelled and calcium can flow in. This results in a higher postsynaptic response than what is proportional to the existing presynaptic signal. Since the cells in the hippocampus have these receptors, they are able to undergo LTP, but only under depolarizations of high enough frequencies. Synaptic plasticity is also impacted by neural stem cells, NSCs. These stem cells are located in the dentate gyrus, DG, of the hippocampus and have been found to generate new neurons. By making new neurons, there are more places for neural connections, and the network of our nervous systems therefore expand rapidly. Neuroplasticity is something we actually rely on, so decreased NSC results in a loss of cognitive function, while increased NSC results in heightened learning and memory. NSCs can be increased through exercise and high learning environments. Essentially, our brains are something we have to work out, and by doing so, they stay sharp. Neuroplasticity from the generation and regeneration of neurons results in dendrites altering themselves within seconds. Neural imaging of such dendrites has shown the creation of new synapses from simply LTP. As the brain generates more EPSPs, the network wants to interconnect more, resulting in the creation of more synapses in the hippocampus. This ends the video on plasticity.